Greetings to all the viewers. Now we are going to discuss about the compound called quinine. Quinine is an alkaloid and it is effective anti-malarial drug. We will discuss about the alkaloid called quinine. Let's go with that. Quinine is a naturally occurring alkaloid used primarily for the treatment of malaria. The main application of the quinine is for the treatment of malaria. It has a long history in medicine and continues relevant today, especially drug resistant strains of malaria. Okay, so even drug resistant strains are present. This quinine is able to work against this malaria. Quinine is derived from the bark of cinchona trees. Cinchona trees, which are the native of South America. From this, cinchona plants are originated, and from this bark we are going to extract the material called chemical component called quinine, right? So this is about quinine. This is a brief info regarding this and overview of quinine. So from where we are extracting? Source: extracted from the bark of several species of cinchona trees, particularly Cinchona officinalis. Cinchona officinalis is the tree. From its bark, we are going to extract the medicinal component called quinine. Classification: quinine is a member of quinoline-alkaloid family. Quinoline means what? Benzo-fused pyridine is called quinoline, and this bicyclic ring system is the core component over there, so that this quinine is the part of quinoline alkaloid, and its formula compresses C20 H24 N2O2. So this is the formula for the chemical component of quinine. Molecular weight of quinine is designated as 324.42 grams per mole. This is about the molecular weight of quinine. Historical significance. Discovery: indigenous people of South America were the first to use cinchona bark to treat the fever. So directly they collected the bark of cinchona tree and they used to relieve the fever conditions in South America, right? Later on, European settled people in 17th century recognized the efficacy of this material in the year 1820. Quinine was isolated by the French chemist. So this is the extraction, right? So initially used by South Americans, and European settlers also recognized the significance, and they also used as a medication from the plant extract. And the French chemist, Joseph Pelletier and Joseph Caventou, these are isolated. So chemical isolation got carried by these two scientists together. So this is about the historical significance of quinine and how can we apply for the treatment of malaria. Let's see. Quinine was one of the first effective treatment for the malaria, a disease caused by the parasites of genus Plasmodium. Plasmodium is the kind of parasite. This is the microorganism which is the cause of disease called malaria, and anti-malarial quinine is essential in order to destroy this Plasmodium. And this plasmodium is transmitted by means of mosquitoes. Anopheles mosquito is able to transmit the disease called quinine. Before the development of synthetic anti-malarial drugs, quinine was the primary treatment. Nowadays, so many type of anti-malarial drugs are available, but quinine is said to be a primary treatment, primary introduced one, right? Mechanism of the action. How it works, we will see about this. Anti-malarial activity. Quinine works by interfering with the parasite's ability to digest the hemoglobin, right? What a parasite will do, what this kind of mosquito will do, mosquito used to inject into the human body and used to suck the blood, right? So whenever we are applying this quinine, so that digestion of hemoglobin by this mosquito will be intervened, and that destroying the ability to digest is possible. Subsequently, it loses the food and it used to destroy this kind of mosquito. So parasite activity will be mitigated so that malaria conditions will be controlled. So whenever hemoglobin digestion is lowered, so that oxygen carrying tendency also lowered, right? This gives rise to the accumulation of toxic substances whenever entire human body will be active and will be fresh whenever continuous circulation of the blood, continuous circulation of the oxygen, continuous circulation of the fluids are possible. Then only entire system will work in the harmonical manner. Whenever this hemoglobin is not in a sufficient quantity, or not in the sufficient amount, what happens? Its oxygen carrying tendency is not that extent, so that all the organs are not properly working, and accumulation of toxic materials are possible. Slowly the destroy of that mosquito is possible. It specifically targets plasmodium parasite such as plasmodium falciparum. Plasmodium falciparum is the parasite, is the microorganism which used to induce the disease called malaria. So that we can mitigate the disease malaria by eradicating this Plasmodium falciparum, which is responsible for clinical symptoms of malaria. So this Plasmodium falciparum is responsible for the disease called malaria. Now, this is regarding what mechanism of action, how it is working. Whenever we are consuming that quinine, so that quinine enters into the parasite, and whatever digestion of hemoglobin is there, that totally resisted. Whenever hemoglobin digestion is not there, oxygen consumption, oxygen carrying will be totally surpassed, and toxic ingredients will be accumulated inside, so that slowly its activity destroyed, and that parasite will be completely removed. This is the way mechanism of action is possible. Now let's go with medical applications. Already we aware this quinine is most importantly used for the curation of malaria. Quinine remains a key drug for treating drug-resistant malaria. Any other drug which is not working upon, at that condition also we can conveniently administrate this quinine as a curative, effective curative, especially when parasites show the resistance to newer anti-malarial chloroquine. Any kind of new drugs if present, at that condition we can go with this quinine. It is often administered in the combination with anti-malarials, doxycycline and clindamycin, and improve and reduce the treatment duration. So individually quinine can be administered and effective treatment is possible. At the same time, it can be blended with some other drugs like dioxycycline or clindamycin. Such materials also can be blended with quinine in order to make it more effective. Now let's go with another application, medical application we are discussing about nocturnal leg cramps. Leg cramps are commonly occurred whenever the person is experiencing severe load upon, or else running very fastly, or else any muscle, it is a kind of a muscle cramp. Muscle, what we can say, relaxation is not that easy. At that condition, cramps are possible, which induce the severe pain over there. At that condition, the administration of quinine is very essential in order to cure this nocturnal leg cramps, though its use in this purpose has declined. So this is the purpose over there. So here a kind of pain which is arrived due to a muscular disorder. So quinine can be applied over there, so that we can use the effective curative agent for these leg cramps. Then lupus and arthritis. Arthritis are nothing but it is a bone related disease, we can say. Whenever joints or ligaments are present at that condition, so whatever softening fluid is there, that totally destroyed, so that bone friction is generated, it used to induce the severe pain. At that condition also we can go with the quinine. Some people use quinine for autoimmune conditions. Autoimmunity is nothing but development of immunity defensive system inside the human organism to avoid any other diseases. That is called autoimmunity. Immunity is used to resist the pathogens or parasites, any kind of disease causing organisms to enter inside. That develop itself, which is called autoimmune conditions, that also generated by means of the drug called quinine. And rheumatoid arthritis are nothing but simply we can say joint pains. So this kind of rheumatoid arthritis joint pains can be mitigated by means of this quinine. So quinine in the tonic water. Tonic water denotes what? That is the ingredient present in quinine is the ingredient present in the tonic water, gives the drink characteristic bitter taste. So this what tonic water contains a bitter taste, and the concentration of quinine in the tonic water is much lower than the medical dosage. It can be conveniently stored as a beverage as well, right? So bitter taste containing water is originated by mixing with the quinine. Now we will go for the synthesis. How can we synthesize? How can we synthesize this quinine? Quinine can be derived from the natural source. As already we aware, Cinchona officinalis as the plant is the main source for the extraction of quinine. From its bark, we are going to produce, and even laboratory synthesis is also possible parallelly. Total synthesis. The total chemical synthesis of quinine is challenging. So because all natural products extracted from the nature are very complicated, and it is very difficult to go for the laboratory synthesis. It's a very complex process. Any natural product synthesis within the laboratory is a complex process, but in order to make the drugs in the bulk quantity, it is mandatory to go for. That's why our pharmaceutical industries are working upon that whatever natural ingredients, plant extracts or animal extract are having that defensive system, and that mimetic compound synthesized in the laboratory as well. And another research with the upgradation of these compounds giving a new kind of materials, or else another route containing compounds. So many kind of ways we have in order to introduce new drugs into the market. Research is going on. If any disease is occurred, so first of all we have to examine the symptoms. Later on, what is the cause for that one? Later on, which is a suitable one. So and in what way we will synthesize. That is the way research is going on in order to cure a different kind of diseases. So quinine synthesis is also a very challenging one. First synthesis of quinine was achieved by American chemist R.B. Woodward and W.E. During in the year 1944. These two scientists, Woodward and During, together worked upon the synthesis of quinine, though it is the complicated process, but they achieved successfully first synthesis of laboratory grade quinine in the year 1944. However, due to the complexity of the molecule, most quinine is still obtained from the nature. So laboratory synthesis is there. So bulk quantity drug, if needed, we have to go for laboratory synthesis, but natural source quinine is also available. Modern use of quinine. While newer drugs like artemisinin and other derivatives have become first-line treatment of malaria, quinine is still used in drug resistant. So Wherever we are consuming the drug, but it is not properly working in order to destroy that kind of disease. That is called drug resistance. That receptors or else that pathogens develop the immunity against the drug which we are continuously consuming. That is called drug resistance. At the drug resistance condition, quinine is the alkaloid which perfectly works upon. So this is the way we can solve the problem by using quinine. Let's go with quinine applications. It also has a role in treating severe malaria. Just now we know severe malaria can be eradicated by this alkaloid called quinine, and it is an anti-malarial drug, and this is the bark extracted from Cincone officinalis. So this is the bark, medicinal value containing bark it will be. Quinine remains historically important, clinically useful drug, particularly in regions where malaria is endemic, right? So here, that particular region, if diseases are spreading, so this quinine is the better alternative in order to go for the curation of this kind of disease. Though it has been largely replaced by more modern drugs, but drug-resistant malaria can be crucially cured by means of this quinine. This is a vital tool and finds a global significance for the eradication of malaria. Now, by this, what we observed, what is meant by quinine, and where we are applying this quinine, and what is the historical background of this quinine, and a quick glance was completed. Now we will go with structural elucidation. Structural elucidation denotes whenever we are taking, for example, Cincone officinalis plant was taken and it is crushed, grinded properly, and you extracted its juice and properly dried and found certain crystals. After getting that crystals, for example, that product, we are not able to know exact structure of that one. There is the systematic literature we have in order to identify what compound is containing inside of that unknown one. So this is the strategy called structural elucidation. In order to elucidate the quinine structure, molecular structure, through the combination of spectral and analytical techniques we are applying. Spectroscopy will be a tool in order to identify the carbon skeleton, functional groups present in that, and the chemical composition can be mentioned with the spectroscopy, and analytical techniques also applied in order to understand the structure of the quinine in general. The structure of quinine was elucidated in the early 19th century, but modern methods offer more detailed insights into the configuration, functional groups, and stereochemistry. Right. So here, 19th century itself, quinine structure is elucidated and its chemical composition skeletal was revealed. But still, there is upgradation and high resolution composition and stereochemistry. What is the arrangement? What is the arrangement of all the functional groups, entire things. Still, any compound or any research never ends. Always there is upgradation further. Any kind of technology, new version is mandatory in order to go for the new innovative methods. That is the reason why, though it is introduced or identified in 19th century, still modern methods are applied and high resolution configuration we are going to analyze for this quinine molecule. Here you can clearly observe quinine is mentioned over here, bicyclic ring system with the nitrogen which is called a quinoline core, and this is another piperidine ring. You can notice again it is a bicyclic one is 6-membered, another is 4-membered ring it will be. This is the way two bicyclic rings are located. Both are joined with the methylene, where you can notice tertiary hydroxy, and another methoxy is also located. And here external, that means exocyclic double bond is there. And this is the tertiary nitrogen. This is the entire composition of quinine, but we don't know. So initially we don't know this kind of chemical composition. We have to go with the step-by-step determination. For that, overview of quinine structure. First of all, elemental analysis reveals that molecular formula is denoted as C20H24N2O2. Molecular weight of this quinine is 324.42 gram per mole, right? Molecular weight is 324.42. And it's a core structure. If you observe the core structure here, two fragments you can notice. One is a quinoline moiety and second is called quinaclidine moiety. This is called quinaclidine. This nitrogen containing one is called quinaclidine. Okay, so quinaclidine is also present. So this is about the core structure of quinoline. It contains tertiary alcohol. Tertiary alcohol means any hydroxy which is present on the carbon which is attached to the two alkyl is said to be a tertiary. And this is the nitrogen containing three alkyl called tertiary amine. Here the functional groups are, one is tertiary hydroxy, another is tertiary amine. This is the way structural elucidation overview we are going with. Now we entered into another segment called techniques used in the structural elucidation. We are adopting machineries, that means analytical tools in order to identify, in order to extract the chemical structure of this quinine. Elemental analysis. Empirical formula of quinine is designated as C20H24N2O2, which is determined using elemental analysis. By this, how many carbons, how many hydrogens, how many nitrogens and oxygens are contributed for this quinine can be determined. This is for elemental analysis. Elements, individual elements can be identified. Mass spectroscopy. Mass spectroscopy confirms molecular weight of quinine, as mass spectroscopy is the device used to identify molecular weight. 324.42 gram per mole is identified. Mass spectroscopy also provides fragmentation patterns. In what way we can go for strategic cleavage of the bonds, and quinquidine rings get isolated from that. So that two applications you can find with mass spectroscopy. Infrared spectroscopy. IR spectroscopy is essential for the functional group identification. Here we can clearly observe tertiary amine is there, alcohol hydroxy is there, and the quinoline, aromatic quinoline ring is there. This identification of tertiary amine with NH stretching, alcohol with the OH stretching, quinoline identified with the carbon-carbon double bond stretching. This is the way IR spectroscopy found the OH is there, amine is there, and carbon-carbon double bond is there. This was identified. Now let's go with NMR spectroscopy, nuclear magnetic resonance spectra. Nuclear magnetic resonance spectra is recorded with the two different instruments at different ppm values, and proton NMR and C13 NMR spectroscopy are used in order to find out, in order to trace out the nature of hydrogens and carbons as well, into the hydrogens and carbon environments to the molecule. Quinine, proton NMR shows the distinct signals. Signals we can characteristic signals are achieved for quinoline and quinoclidine. So quinoline and quinoclidine, both the moieties we can clearly observe. This is called quinoline and this is called quinoclidine. So both the kind of structures are clearly identified by proton NMR. Characteristic peaks of aromatic quinoline aliphatic chain of hydroxy OH hydrogen is easily identified. Amine hydrogen is also found. So this is clear NMR spectra which we can see for this quinine moiety. And ultraviolet spectroscopy. So here wavelength and absorption were taken for the graph plotting, And ultraviolet visible spectroscopy of quinine has a conjugated system due to aromatic quinoline ring and distinct absorption bands in the UV region. Here, always UV peaks are not that sharp, and broad peaks were appeared over there, and these are used to identify conjugation. The purpose of UV visible spectroscopy in order to trace out the presence of conjugation, annular systems, whether present or not. So this is the way we can go for the analysis. Methyl, how many methyl groups are present? So this is the purpose of UV visible. So UV visible spectra of quinine pertaining are provided on the screen. This technique helps to confirm the presence of quinoline moiety. Based on this, we can clearly observe quinoline is present within the compound. Now we entered into another segment called X-ray crystallography. All these are the analytical tools only, proton NMR, ultraviolet, IR, UV, all these are called X-ray is also called, all these are analytical tools for the identification of unknown compounds. X-ray diffraction studies provide 3D structure of quinine. X-ray diffraction is mainly used for 3D structure analysis, spatial arrangement of the atoms and conformation of bicyclic quinine system fused in the quinoline ring. X-ray studies are provided over energy in centimeter inverse are plotted against the relative intensity values, and this kind of curvatures and this kind of graphical representation we can clearly observe. And X-ray crystallography reveals absolute configuration. Absolute configuration denotes what? Any compound, either S configuration or on R configuration, if you are tracing out, that is also called as absolute configuration. It purely depends upon stereochemistry of the molecule. And stereochemistry can be extracted whenever chiral carbon atoms, chiral centers are analyzed. Conforming quinine exists in diastereomeric when. So two quinoline molecule, two quinine isomers are existing in the diastereomeric relationship. Specific stereochemistry at its chiral carbon atoms. Quinoline contains two chiral carbons. Here, one chiral carbon with OH, H, and this heterocyclic ring, and this is heterocyclic ring. And this is another chiral carbon with CH3H. This side one type. This is another type. Chiral carbon denotes any carbon with 4 different functional groups is designated as a chiral. So that they used to give 2 chiral carbon atoms, and RS configuration can be calculated at these 2 points. And they only responsible based on the orientation of the functional groups around the chiral carbon, that only imparts the biological activity. Biological activity purely depending upon orientation of the functional group, where they are locating. Key structural features of quinine. So quinine structural features are identified quinoline ring. So here is the quinoline ring. Benzene fused with the pyridine is called a quinoline ring. This quinoline moiety is aromatic heterocyclic ring. Aromatic heterocyclic ring it will be, and it is fused with the benzene pyridine. It is the benzo fused ring. Benzene pyridine fused ring it will be. This is responsible for anti-malarial activity. So this moiety core for anti-malarial activity, and quinine ring in the sense wherever piperidine ring which is bridged with another cyclic ring which is called quinine ring. So this is a bicyclic ring, and where you can find a tertiary amine. This tertiary amine containing quinine ring is responsible for rigidity of the molecule that used to fix the molecule. So biological function is probed by means of this rigidity. And chiral centers, as we are aware, wherever 4 different functional groups are present, and these 2 pink lobes denotes these 2 are the chiral centers over there, and which are responsible for the activity. That means a potency, the molecule potency will be altered by means of its orientation. Functional groups. It contains a tertiary amine and a hydroxyl group attached to the side chain. Tertiary amine we can clearly notice, and hydroxy is also present. These are regarded as functional groups located within the molecule. And it is concluded that structural elucidation of quinine involves combining several techniques. So many techniques we gone through. Elemental analysis we did, IR spectroscopy we adopted, NMR spectroscopy, UV visible spectroscopy, X-ray crystallography. So many techniques we used in order to identify the exact chemical composition of this quinine alkaloid. And exact geometry, RS notations, absolute configuration, it's a stereochemistry. Entire concept was well demonstrated. Quinoline and quinacridine rings along with the chiral centers and the functional groups identified using NMR, IR, MS, UV visible, X-ray, crystallographic techniques. And it mentions that quinine is a natural anti-malarial agent and is specific in the stereochemistry and effective drug it will be. Now, by this, we gone through whatever product are juices extracted from the plant, cinchona bark. Now we gone through the structural elucidation. The next segment we are discussing about synthetic preparation of quinine. As already we are aware that Woodward and Doering are the scientists together initially did the first synthesis in the laboratory, and how can we go with that? So many steps are available, so many steps are present over for this synthesis of quinine. So that we are simplifying in the certain extent in order to give the convenient pathway. Right. So initially we are taking the starting material called 7-hydroxy isoquinoline. This is called 7-hydroxy isoquinoline molecule. Formaldehyde and piperidine was treated. Formaldehyde used to give the CH2OH at this position. CH2OH will be generated, and whenever piperidine added, saturated pyridine is called piperidine, which is added to this CH2OH, so that that 6-membered ring is added. Methanol and sodium ethoxide, when added, the entire piperidine will be removed and it will be reduced to give methyl. Simply by this entire process, whatever isoquinoline core was taken, the methyl is substituted at the position 6. After getting this one, multiple steps, multiple reagents were treated one by one, step-by-step manner, so that we are going to achieve that saturated ring system. Let's see one by one. Adams catalyst in the acetic acid. Adams catalyst means what? Platinum oxide in a water. This combination, platinum oxide in a water, is called Adams catalyst. This is used to saturate heterocyclic ring. It is very specific for the heterocyclic ring reduction. Later on, acetic anhydride in a methanol was treated. Acetic anhydride in methanol is nothing but acylation on this nitrogen atom. This is the way we are going to protecting group it will be. Later on, Rainy nickel in the ethanol was treated. Rainy nickel ethanol used to reduce the next segment, that is another aromatic ring system. Chromic acid and acetic acid. Chromic acid is a mild oxidizing agent used to convert hydroxy into ketonic functional group. These many steps we are going with. First of all, we are going for the reduction of heterocyclic ring with the Adams catalyst, and acetic anhydride in the methanol used for acylation in acylation process, Friedel-Crafts acylation. Rainy nickel in the ethanol used to reduce the another annular ring, aromatic ring, benzene ring, and the chromic acid, acetic acid used to convert OH into double bond O. This is the way a compound will be generated. Later on, alkyl nitrite. This is called ethyl nitrite. Ethyl nitrite, when added, that used to bind upon this molecule. Further, platinum in the acetic acid were treated. So what happens? One of the cyclic ring will be opened, and we will get what? One side carboxylic acid, another side double bond will be generated. This is the ring opening reaction. Right. After getting this molecule can be rearranged and resegregated in another manner as well. We can write the structure in another. possible way also. So this is the piperidine ring, where you can notice this is the vinyl moiety. Double bonded carbons will be there, and on either side we have the carboxylic acid chain. This can be rearranged in simply we are going to change its orientation, right? Alkylation with iodomethane. Iodomethane is used to quaternize this nitrogen. This nitrogen is a secondary over there. Whenever alkylation get carried, what happens? Nitrogen is accompanied with the 2-methyl, and it will be quaternary nitrogen. And so many other steps also carried in order to reduce the complexity of the entire process of synthesis. We mentioned with the double-headed arrows. And further, quinoline core is also joined with this, and we are going to achieve this quinoline moiety is in contact with this ketone, and here we have another side chain with this piperidine, and exocyclic double bond is also there. And next, whatever quinotoxin was synthesized, further process was carried by Rabe and Kindler. Rabe and Kindler were extended the research further, and finally we achieved the final compound called quinine. Quinine is represented as quinoline with methoxy, and here hydroxy, and this is quinqualidine ring, and this exocyclic double bond. The entire system was provided over, and this is the one called Woodward during formal synthesis, right? So this is laboratory grade synthesis for this molecule. We are going to start with isoquinoline skeleton. So many multiple steps got applied, and we will get quinine as the final product. Now we will go with the application part. Quinine contains wide range of applications, primarily in the medicinal field due to unique pharmacological properties. It is most abundantly valuable and most widely used type of chemical worldwide. Historically, most famous for its role in treating malaria, anti-malarial drug quinine will be. But it also has other therapeutic use in the industrial applications. Primary application, quinine most significant in anti-malarial drug. First effective treatment of malaria. Plasmodium falciparum is the parasite which induces this disease called malaria. This quinine, whatever we are administrating, we are consuming, that used to destroy Plasmodium falciparum parasite, the most dangerous and drug-resistant malaria parasite. Mechanism, how it works? It used to break down hemoglobin. Hemoglobin circulation will be stopped. Hemoglobin circulation, so that oxygen circulation stopped, so that the toxic chemicals developed within the parasite. Yesterday, although newer anti-malarial drugs are available, but it is working upon drug-resistant malaria therapy. This is effective over there. Severe malaria can be destroyed. And one more important point I would like to introduce. In Hyderabad, Sir Ronald Ross is the scientist. He researched upon malaria disease. What is the malaria disease? What is the cause of that one? How to reduce the symptoms of that malaria by using this quinine. That entire research got carried in Hyderabad by the scientist called Sir Ronald Ross. So that Ronald Ross anti-malarial, that hospital will be located in that Hyderabad. Treatment of nocturnal leg cramps. Leg cramps easily, effectively mitigated by means of this quinine drug. Lupus and rheumatoid arthritis. So rheumatoid arthritis are nothing but wherever all the bone joints are connected with the ligaments, where synovial fluid will be there. Synovial fluid is the lubricant in order to slide one bone upon another whenever we are rubbing, whenever we are walking, or else doing work with that. So synovial fluid is the kind of device which used to slide one bone upon another. whenever that fluid got completely evaporated, so bone friction is possible that used to induce the severe pain that wherever joints are present everywhere, joint pains are possible, that is called water rheumatoid arthritis. There's, in that condition we can conveniently apply this quinine as a effective drug. It is less commonly used in conditions today, and largely quinine, other therapies also available, but quinine is effective against that rheumatoid arthritis. Antipyretic and analgesic. So the the medicine which used to cure the fever conditions is called a antipyretic. The drug which used to reduce the pain conditions is called analgesic one. So antipyretic and analgesic are the properties which are present for this quinine. Fever reducing tendency will be the pain relieving tendency. This is another application we can notice. In tonic water, tonic water denotes what? Quinine used in the tonic water today. Lower the medicinal dosage, say for the regular consumption as a beverage. So tonic water is bitter in taste. Medical dosage can be minimized by means of the consumption of this tonic water, and it is regularly applied in the beverages. Cold storage devices are called beverages. Light sensitivity. Quinine is sensitive to UV light, which causes the fluorescence. Whenever quinoline is direct contact with the light, what happens? It used to reflect that light, so that fluorescent tendency will be there, creating the bluish glow. It will glow like a blue color one. So this is the kind of light sensitivity. Quinine is the key ingredient in the tonic water. It imparts the characteristic bitter taste. Tonic water is having bitter taste, and tonic water originally consumed as a prophylactic against malaria. So this tonic water is very effective for the treatment of malaria. And as we already were, whenever it is in direct contact with the sunlight, like ultraviolet radiation, it used to reflect that light, so that it contains a fluorescent property. Because of fluorescent property, we can utilize as a fluorescent markers. In electrical chemistry, quinine natural fluorescence UV light makes useful as a fluorescent marker in the chemical analysis for the scientific researchers. So these markers are composed by quinine, which is in contact with UV light. Applications in the spectroscopy. Fluorescent properties allows to use the fluorescence spectroscopy. Calibration standard and probe the experiments requires the tracking of the molecular interactions. Being it is a kind of light reflecting kind of material, fluorescent a glowing material we can say, which is used as a calibrating standard in the spectroscopic techniques, so that tracking of molecules is the most convenient with this fluorescent marker. Potential antiviral activity. There has been interest in investigating quinine as its derivative for potential antiviral properties. Quinine contains antiviral property. Applications are still experimental. So antiviral properties are underway for eradicating the viral related diseases, but it is going on. Some research has explored whether quinine might have activity against certain viruses, although it is not currently used as a standard antiviral. So the process is underway. That's why we are not supposed to say it is a standard drug in order to cure the diseases caused by the virus. So in the near future, we are going to experience the quinine as a potential antiviral agent as well. Molecular template in the organic chemistry. Quinine has the derivatives. Quinine and its derivatives are used in the chiral templates. So chiral templates are developed by means of this quinine and ligand in the asymmetric synthesis. So by extending its electron releasing groups, so that it is working like a ligand, and asymmetric synthesis, we are going to apply them. Its chiral centers are exploited to produce the molecules with the specific stereochemistry. Stereochemistry Already we aware two chiral centers are located in this quinine molecule, and whenever these chiral centers are properly investigated, so we can identify, we can trace out specific stereochemistry of the molecule, making it more useful in the environmentally pure compound synthesis, especially in pharmaceuticals and as well as industries. So by by exactly analyzing what is the orientation of all the functional groups over there in the pharmaceuticals and in the industries, we can find more applications for these quinine molecule. Cinchona tree cultivation and quinine derivatives. As we already aware, Cinchona officinalis is the plant from which whose bark we are going to extract this quinine alkaloid. Quinine is the precursor to several modern anti-malarial drugs. Nowadays, so many new drugs are coming. For the development of new drugs, the starting material might be quinine, because it is the first generation anti-malarial. So that by using this one, we can extend its chemical composition, so that new level of anti-malarial drugs will be introduced. The cultivation of cinchona trees, the natural source of quinine, remains important in obtaining both quinine and its derivatives. Quinidine is also used in erythematic agent in the treat of heart conditions. This is another analog for this quinine. This is the way cinchona tree cultivation is more important in order to extract cinchona from the natural source in the maximum extent. By this, we concluded the session. Hopefully, the entire session is very informative, and this is the content related with the alkaloids. We already started the series of natural products, alkaloids and terpenoids. Introduction class was done, morphine completed, and now we entered into quinine. So where the entire comprehensive description is provided for this chemical entity as an anti-malarial agent. Thank you very much for your patient listening. This is Dr. Kavita Kottireddy Kota. Thank you very much.